When you try to do this experiment in animals, you have to inject them with a radioactive material so that you can then look at that radioactive material. And that's what some of those pictures were in the neurogenesis. You had to look at that radioactive material on a slide. So how do you think you do that? You inject them with a radioactive dye, basically. It is incorporated into new cells. It goes to the DNA, it's replicated, it goes into the new cells. You let them live for a while so you think that they've had some new nerve cells form, and then you kill them. And you slice up their brains and you see. Well, you can't really do that experiment in humans very well. So, Dr. Gage said, is there any way that we can approach this where there might be some humans who already have radioactive dye who would allow us to have their brains at autopsy? And he realized that for people with a particular kind of cancer, it happened to be throat cancer, squamous cell cancer of the throat, that at that time that he was doing his research, people who had that particular kind of cancer received a radioactive dye injection as part of diagnostic procedures. It was standard practice, it wasn't extra. And he said, you know, here's a kind of natural experiment. Here are people who already have radioactive dyes being administered. We're not asking anybody to do anything dangerous. But it's going to be a difficult conversation to have with people about having their brains at autopsy. And so he went about thinking about this and talked to one of his colleagues who happened to be a clinician researcher. He said, you know, you'd be amazed at the generosity of human beings in advancing the knowledge for people for generations after them to come. I don't think you're going to have trouble at all. And so indeed, he went to a particular cancer research center. He spoke with people about the nature of his interest, the nature of his experiments, and asked simply that when they died, would they be willing to have an autopsy, and could he have their brains? 5 people said yes. And by virtue of their gift to the rest of us, because that's the way I think about it, and it's the way Fred thinks about it. By virtue of their gift, he was able to demonstrate that, lo and behold, they had those radioactive dyes present in the hippocampus. Just like the songbirds, rodents, shrew monkeys, tree monkeys, rhesus monkeys, that we have in common the capacity to repair our brains, to make new brain cells, to regenerate neurons. And this was huge. He continues to do all kinds of work, and now also directing some of his interest towards looking at people with Parkinson's disease, Alzheimer's disease, even looking at people with depression. I'm going to say a word about that in a moment, because people with depression have shrinking hippocampi. So the human story we were told by Fred. But what do you think you ought to be ready to challenge me with? Or maybe anybody would like to challenge me. Because here's what you ought to be wondering. That's great. So you grow some new cells. Do they work? Are they functional? You know, you have a cut on your arm, your skin grows, makes new skin, covers up that scar very nicely. But a lot of people get keloids. Skin just keeps growing and growing, and you end up with a big fat scar. They're not good for anything, they just grow. So just growing isn't enough. The next part of this story had to be, are they good for anything? It's great that we make them, but do they work? So back to the animal studies. And this is where it gets, I think, for us, really fun. What Fred and many, many others have been able to demonstrate is that rats, mice, who live in enriched environments have heavier brains. That means they have more brain cells. They have more synapses. Synapses are the connections between brain cells. They have more branching of the dendrites. I'm going to have a picture for you in just a moment of a dendrite. And most importantly, they learn better. They learn more, they learn better, they retain longer. And this is true even for very old mice. This is not just true for young mice. It is not just true for young adult mice. It's true for old mice. And in a minute, you're going to know that it's true for old people too. Now, what is, I think this should be my next slide, what is an enriched environment for a rat or a mouse? Well, it's kind of funny because it's the same thing that's enriched for us, or maybe it's because it's the only way we know how to enrich their environments. But enriched environments have to do with toys and friends. It has to do with having more mice to hang out with. It even has to do with how big your cage is and how many toys you can get in it. Not exactly a great message for somebody who's kind of anti-consumer, but it may turn out that he who has the most toys does win. I don't know. But an enriched environment is about having stuff to explore, stuff to do, stuff to play with. And that's really important, that this has to be embraced, or you get the most benefit when you embrace it with a light touch. When it's playful, the way kids play at learning. You watch the pleasure and the joy that kids experience when they learn something, and you'll know what I'm talking about. It's fun. It feels good to solve problems. It feels good to have a knotty, thorny issue you can't quite get. It feels good to memorize a poem. It feels good to do it with others. It's good to play at learning. So we know that enriched environments, play, and friends. We know also that if mice, at this level, mice are trained to run a maze, that they get a lot of practice trials, that if you put like new little barriers in their mazes and really stimulate their learning, you make them learn more than one maze and so forth, they also have heavier brains, more synapses, more dendrites, better learning. So not just learning their one maze, but playing. And then finally, as an independent variable, meaning that if you didn't do anything else but run on that wheel, you got the same benefit. Physical exercise cannot be overestimated for its importance in healthy aging. It absolutely can't be for us, rats, monkeys, and interestingly, crayfish. Somebody's actually found a way to get a stimulating environment for crayfish. And I thought I had a picture, but I guess I don't, so maybe I will in my next session. It's wild to me how many other species people look at. That's what stimulates neurogenesis. That's what stimulates new brain cells. Now, oh, I do have the picture. There it is. There's my crayfish picture. Okay. Not only do we know how to stimulate this in a positive way, this being neurogenesis, but we know what stops it in its tracks. There are two things across all animals, including us, that have been studied that have demonstrated quite effectively how to stop the brain from making new cells. And they are, first of all, isolation. If you take this poor little crayfish, or maybe you say crawfish here, but crayfish, and put it in a tank all by itself, with only one little tube to swim in and out of. I guess they must like some periods of dark. That's not, I don't know why that little tube is in there, but that's how they always do it. One little tube, nothing to do, no friends, no little plants, in there by itself. The hippocampus of that crayfish shrinks. If you look at all of these crayfish,